Hi, my name is Lauren Butler. I'm in Dr. Zoval's Chemistry 108 class, and I'm doing a presentation video on yeast fermentation. Yeast is a non-filamentous unicellular fungus that are generally shaped as an oval or sphere. Yeast is part of the eukaryotes, which includes fungi, algae, protozoa, and helminths or worms. The fungi group can then be divided into three types: yeast, molds, and mushrooms. Fungi degrade organic material into inorganic materials and play a role of recycling nutrients in the environment. Yeasts are broadly found in nature on fruits and leaves in the form of a white powdery coating. They are single cellular and reproduce through budding or fission. The oxygen requirement for yeast is a facultative anaerobe. This means that yeast have adapted the ability to grow with or without oxygen as a source. Without oxygen, yeast grows using fermentation or anaerobic respiration. Yeast has the presence of enzymes catalase and superoxide dismutase that allows toxic forms of oxygen to be neutralized and thus can use this form of oxygen. This adaptation of yeast allows for growth and survival in many various environments. An example of a facultative anaerobe bacteria is E. coli. This experiment uses the materials: a packet of yeast, available at any grocery store, a small plastic water bottle, 16 ounces or smaller, 1 teaspoon of sugar for fermentation, some warm water, and a small balloon to collect the byproducts of yeast fermentation. The procedure includes the following steps. First, add about an inch of warm water to the plastic water bottle. The second step is to add the entire package of yeast. And swish for a few seconds. The size of the bottle is important for diffusion of carbon dioxide. A larger container will inhibit the rate of diffusion. The third step is to add the sugar and swish the water bottle again. These two steps start the fermentation process. The fourth step is to expand the balloon by blowing it up a few times. This helps stretch the balloon out in the process of diffusion of carbon dioxide byproduct into the balloon. The last step is to place the balloon on the top of the neck of the bottle and wait about 20 minutes and the balloon will inflate. One key to the procedure is a warm environment. This helps the chemical reaction accelerate. The end result is glucose fermentation by yeast with a byproduct of anaerobic respiration as carbon dioxide. The yeast consumes the glucose and ferments the glucose to produce energy in carbon dioxide, which blows up the balloon, as seen as the byproduct. First, the gas enters the balloon, and once the bottle is filled up with carbon dioxide, the gas then diffuses into the balloon. An example of yeast used in everyday life is in the bakery to make bread. The holes produced in the bread after baking were a product of tiny bubbles created through yeast. After leaving the oven, the yeast lyses, and what remains are holes created through yeast. Although this experiment uses Saccharomyces species as a safe form of yeast, not all yeasts produce positive reactions. Other yeasts, such as Candida albicans, is an opportunistic pathogen what is normally found in the human normal flora of the body, but in certain cases can be pathogenic or harmful to the body. Candida albicans can overgrow and thereby producing thrush in the oral cavity of the mouth or overgrow in the intestinal tract or genital area. HIV infection, diabetes, and the use of broad-spectrum antibiotics increases the risk of this infection. The ability for yeast to ferment sugar such as glucose, is an anaerobic activity and is not only used to blow up balloons, make holes in bread, but is capable of creating a very much consumed product by many, beer and wine. Saccharomyces cerevisiae produces alcohol as a byproduct in the fermentation of grapes and therefore helps to create wine. Yeast ferment sugars, but not all in the same way. Two results can be carbon dioxide or ethanol, such as in alcoholic drinks. Therefore, another experiment that can be done at home is the creation of wine using the yeast culture Saccharomyces cerevisiae with mashed grapes and incubating at a temperature around 30 degrees Celsius for at least a week. However, it is recommended to use the pasteurization technique by boiling the mashed grapes in a test tube or glass container for at least 5 minutes in order to remove other natural yeasts found on grapes, therefore resulting in a better wine and a controlled fermentation process. Differences between yeasts and molds and mushrooms are that yeasts are single-cellular and facultative anaerobes, while molds and mushrooms are multicellular and obligate aerobes. Therefore, need oxygen to grow as yeasts do not. Yeasts also thrive in environments with a pH slightly lower than 5.6 and high osmotic pressure. This helps separate bacteria and yeast using a specific agar in a laboratory setting called Samburan dextrose agar. The fermentation process in itself is extracting energy, such as in the chemical reaction output of carbon dioxide with yeast, from the oxidation of organic compounds. In anaerobic bacteria or fungi, when oxygen is not available and when oxidative phosphorylation is not available to produce ATP for energy through glycolysis, as seen in the chart right here, and growth, which is highly important characteristic. However, yeast, due to their characteristic as a facultative anaerobe, is still able to use oxidative phosphorylation to produce energy in conditions with or without oxygen, as seen in the fermentation process. A chemist, Louis Pasteur, who developed the pasteurization process to prevent spoilage of beer and wine, as well as the sterilization of food, such as milk, in 1864, was also the first person to demonstrate through experiments the action of yeast fermenting glue into ethanol along with carbon dioxide in 1857. This was part of the golden age of microbiology. He also explained that glucose converted into puric acid under normal circumstances, with oxygen enters a series of chemical reactions and then directly enters the respiratory chain, where 36 to 38 molecules of ATP is produced for each molecule of glucose oxidized. Without oxygen, such as anaerobic conditions, puric acid follows two pathways dependent on the cell type, as seen on the chart. One puric acid is produced into ethanol and carbon dioxide, such as yeast, and two converted into lactate by the lactic acid fermentation process. The second pathway occurs in some bacteria. Humans metabolize puric acid into lactate due to a lack of enzymes needed for alcohol production. Heat is also important for fermentation. In cool environments, it is much harder for yeast to ferment, and therefore the speed of reaction occurs at a slower rate. Heat speeds up the chemical reaction occurring. Many chemistry topics are related to this experiment. One is the diffusion of molecules from a large area to a small area. Two, chemical reactions are directly related to temperature. Blue was started with cold water, and therefore it's taking a longer process. 3 is the process of converting glucose by yeast into carbon dioxide through the fermentation pathway. This concludes my experiment. The balloons right here, shown red, that I just performed for you, these all have been done. Blue used cold water, and there's the chart right there. Thank you so much for listening to my presentation, and I hope you have a good night.